Welcome everybody to our M2M new format, new event at BioRen. M2M stands for member to member. So we are here to showcase our members to other members. My name is Annalisa Zuccotti. I'm in charge of communication and events at BioRen. And today I came to Ladenburg at Heidelberg Pharma. And I'm here with Artyom. Please shortly introduce yourself.
Thank you very much, Annalisa. It's great to be here and to have the opportunity to present our company to the audience. So my name is Artyom Vishnev. I'm Associate Director Business Development here at Heidelberg Pharma. Actually, I'm scientist by training and still a scientist in my heart. I studied biotechnology at Technical University Darmstadt, done my PhD at the University Hospital in Heidelberg. After that, I joined Novartis for a couple of years, and 2018, so to say, back to the roots, joined Heidelberg Pharma as a group leader in a bioanalytical department. And since 2020, I'm part of the BD team.
Great, thank you. So we are also happy that you're back to Heidelberg and after your little tour through Europe. So while we were preparing this event, so we thought about the name M2M, and Stephanie and I, my colleague in communication, we continuously thought about M&Ms. So we decided to start the interview with the icebreaker. So I brought here 2 jars, one with M&Ms, one with gummy bear. You can keep one, so be careful in your decision.
I would definitely prefer M&Ms.
M&Ms, perfect. So here
with peanut butter.
Only peanuts. So peanut butter was impossible to find here, but good tip for the next meeting. Great. So Artyom will be able to keep that. But actually, when we arrived here this morning, we asked the same question to your colleagues. So we have now a very short video to give you an impression about Heidelberg Pharma. And also we are curious to see if your colleagues have the same taste. Thank you, Stephanie, for starting that.
Hello, this is Stephanie from BioRN, and today I'm right on site at Heidelberg Pharma for the M2M event. But before we start, I would like to show you around and give you some insights. And I'm really very curious whom we're going to meet on our way now to the conference room. So are you curious as well? You're going to follow me? Okay, so let's go. I think here's somebody waiting for me. Hello, hello.
Yes, hi.
Hi.
Hello.
Hi, I'm Stephanie from BioRN. Nice to meet you.
Nice to meet you. My name is Anniko.
Nice to meet you, Anniko.
Hi.
I have some energy sweets with me. Do you like or do you prefer M&Ms or better Haribos?
So definitely M&Ms. My favorites are those with peanut butter. They're really delicious.
So the energy is with you. Okay, Anniko, what are we going to do now?
So I take you upstairs to the lab space.
Okay.
And there you can see what we are doing here at Heidelberg Pharma.
Great, so let's go. What do you do at Heidelberg Pharma?
I'm the head of the biochemistry and cell biology department at Heidelberg Pharma, and in my department we are performing the conjugations of the antibodies to the payloads, and we make the characterization and also testing the ADCs for their efficacy on cells.
Okay, sounds very interesting.
It is very interesting. And now, yes, I bring you to the bioanalytical department, where the analytical characterization of the ADCs are performed.
Okay, very good. How many people work here at Heidelberg Pharma?
So I think so, approximately 120 people.
Wow.
Quite much people, yes.
Okay.
We have here a chemistry department, the biochemistry cell biology department, the bioanalytics, and also the clinical team and CMC and many different other departments.
Great. So I'm really curious to see now the lab space. We go? Okay, let's go. So let's go. Door is gonna open, and we're right inside.
So we are now here in the floor of the bioanalytics.
Okay.
And here I think, or was it there?
It should be this room, I guess, I suppose.
No.
No? Let's have a look. Maybe the next door.
Where we will find her. Here she is.
She's there. Okay, I'll come. Hi, hi, hi.
So,
This
my
is-
colleague, Marisa.
Hi, Marisa. Nice to
Hi.
meet
Hi.
you.
Hi.
Great. This is the lab space. And Marisa, what are you doing here?
Hi, I'm Marisa. I'm the director of the bioanalytical department,
Okay.
and we are characterizing and analyzing our antibodies, our payloads, and antibody drug conjugates in biological matrices. And therefore, we are developing our own bioanalytical methods, mainly immunoassays as well as HPLC mass spectrometry-based assays. And now you will meet my colleague Arthur.
Very good. Thank you very much. But before we close this video, a question for you. What do you prefer? BioRN M&Ms or BioRN Haribos?
Haribos. I take the Haribos.
So the BioRN energy is with you. Thank you
Thanks a
very
lot.
much, Marisa. Bye-bye.
Thank you very much, Stephanie, and happy that the energy is with Heidelberg Pharma now. Great, thank you very much. So now we have literally seen Heidelberg Pharma. We can start with some questions. So maybe you would like to tell us shortly the story of this company. When was it founded? By whom? What was the motivation of the founders and so on?
So actually, the history of Heidelberg Pharma goes back to two companies. The Vilex, it's a Munich-based, has been a Munich-based company, and Heidelberg Pharma Research, its subsidiary. And Vilex was founded in the later 90s by a group of scientists and medical doctors from the Technical University Munich, and become a publicly listed company around 2000. The Heidelberg or Ladenburg-based subsidiary was a spin-off of Boehringer Mannheim, and the scientists here were focusing first on small molecules and later on developing ADCs. After Vilex was restructured around 2010, 2012, the main office has been moved to Heidelberg, to Ladenburg, and the company was renamed to Heidelberg Pharma. We have from this times, we have a legacy portfolio that is out-licensed to different partners around the world. For instance, Telix Pharmaceuticals in Australia or Red Hill in Israel.
Okay. Perfect. So as you can see, I'm starting asking some questions to Artyom, and I have a long list, so it will be a good half an hour, but obviously you can post also your questions in the chat, and we will select 2 or 3 of them to ask him later. So we said we keep going, so maybe you can tell us about the major efforts and development of the company since the foundation.
I will start from the restructurings around 2012. So after this restructuring, the entire company started focusing on developing novel, innovative antibody drug conjugates with a really unique mode of action. And maybe I can ask one question back. Maybe you're a mushroom picker?
No. My in-laws are mushroom pickers, and then I cook and eat them only.
Okay, but maybe someone in the audience are familiar with the so-called green death cap mushroom. And our technology, our platform technology, is actually based on the toxin amanitin that has been identified many, many years ago in this poisonous mushroom. There are 2 major milestones I would like to mention. First of all, we have established total synthesis of amanitin derivatives that we are using for our ADC platform. So we are not collecting mushrooms from the forest.
Okay.
And the second very important milestone was bringing the payload amanitin into the clinic. So we're currently in phase 1 clinical trial with our lead candidate HDP-101, which is targeting BCMA for treatment of multiple myeloma.
Okay, perfect. And what is the mission of the company today? So you gave us this nice overview on the journey, but what is today the mission?
Very good question. Of course, the mission of the company is to provide new options in cancer therapy to the patients. We call our technology ATAC technology, and this platform technology enables us to produce potentially effective treatments for a really wide range of tumors, with the ability to overcome tumor resistance and to eradicate dormant tumor cells that are, how we believe, involved in the disease relapse. We also have, we're developing a platform-wide specific biomarker that potentially would help us to identify patients whose tumors are more vulnerable to treatment with ATACs. This whole technology, ATAC technology, is like an engine that allow us to build a rich proprietary pipeline of potentially best-in-class drug candidates for both hematological and solid tumor indications.
Okay. And is something changed also along this journey that you just described?
Indeed, yes. Over the years, the company has developed a really extensive expertise and extensive patent portfolio around this compound, amanitin. We all in the company are super proud that we are the first in the world, to our knowledge, that has brought an ADC with the payload amanitin into clinical development. And over the last couple of years, the company has experienced a constant growth. And our current goal is to become a global player in the ADC field. The current strategy is to validate the technology platform in clinical trials, to broaden its application based on its mode of action, and use the technology to develop new therapeutic options for patients. We have a hybrid business model. So I've mentioned before, we have our own proprietary pipeline, which we constantly fill with new candidates, but we also out-license our technology to partners in pharma and biotech around the world.
Okay, perfect. So I think we will come back later to this out-licensing eventually. We've heard about the foundation, the history, this journey, and the new consolidated mission at Heidelberg Pharma. So now, if you would use few words, how would you describe the unique selling point of this innovation made at Heidelberg Pharma? So what makes your company innovative?
I will try to do it in few words, yes. So our therapeutic approach is the inhibition of RNA polymerase II. And this is a mode of action that has never been used in the clinic before. By targeting such a fundamental cell function like the mRNA synthesis, allow us to potentially to target untreatable tumors. And I mentioned before this dormant tumor cells, which are the cause for tumor relapse. We believe having the biomarker in our toolbox makes it to a very powerful tool to identify patients who would likely benefit from treatment with our ATACs.
Okay, perfect. So thank you for keeping the promise and use a few words. But now I give you also the opportunity to dig into the science behind that. And I mean, the you that you are listening, most of you are scientists, and so I think they will enjoy also if you, if you tell us about the scientific approach behind that.
Of course. So, as I said, to our knowledge, we are the first company bringing amanitin and this mode of action, the inhibition of RNA polymerase II, into the clinic. And there's quite a simple reason for this. There's only one specific inhibitor of RNA polymerase known to man, and that's amanitin. But its toxicity prevented its usage for treatment of patients. And what Heidelberg Pharma has done over the last couple of years, our great researchers here have engineered a therapeutic window to amanitin and enabled its application for patient treatment. Speaking as a scientist, amanitin is a beautiful molecule. It's a bicyclic octapeptide, which is super hydrophilic. And this hydrophilicity differentiates it also from payloads that are currently in development or already approved in the clinic. And this hydrophilicity makes it to a perfect payload to target resistant tumor cells. One of the potential resistance mechanisms of tumor cells is upregulation of so-called efflux pumps. And you can imagine these tumor cells like a bucket with a hole. So you pump a payload or a toxin inside the cell to kill the cell, but the efflux pumps just pump the drug out. So this cell can survive the treatment. Mm-hmm. But being so hydrophilic, amanitin is not a substrate to those efflux pumps and can overcome this resistance mechanism. I have mentioned several times already this ability to kill dormant tumor cells, or so-called cancer stem cells. The reason is also quite simple. So even though those non-dividing tumor cells need some kind of housekeeping transcription, they cannot just shut down the RNA polymerase or the mRNA synthesis. But by targeting such a fundamental cell function, we can even kill those cells. And another beautiful story I would like to tell, and it's actually really beauty of the science. It's the biomarker story. So we found this potential biomarker just by serendipity. During collaboration with MD Anderson Cancer Research, the researchers there found that some tumor cells are more sensitive to treatment with amanitin. Those cells were so-called 17p deleted cells. So tumor cells often delete a sequence of chromosome 17, where the TP53 tumor suppressor gene is located to become more aggressive. And really, just by serendipity, we found that another gene is in the close proximity to TP53, and this is POLR2A. POLR2A is a gene that is encoding for a major subunit of RNA polymerase II, so the intracellular target of amanitin. And by deleting TP53, POLR2A is co-deleted in over 95% of cases. So what happens? These tumor cells have less RNA polymerase in the nucleus. So at the end, we need to bring less molecules of amanitin inside those cells to drive the cell into apoptosis. And from my point of view, it's a real beauty of science.
Yeah, thank you. Perfect. This is really what we wanted to hear about the beauty of science, and we see also your commitment and enthusiasm in telling that story. So today we are here in a bigger context. So Heidelberg Pharma is one of many biotech company here in this innovative ecosystem. Heidelberg Pharma has been since the very beginning, since its foundation, member of BioRN, and so I've checked it. So it was the early 2000, and yeah, so you have the opportunity now. So members are listening to you. So how can you help other members? So what can you offer them?
I believe, as you said, we are members since more than 20 years, early 2000s, and being more than 20 years in the biotech business, we definitely could share our extensive experience and knowledge with startups in our BioRN region.
Perfect, thank you. And I mean, in a network, so a network means belonging and exchange. So let's look on the other side. So how can other members support you? So which kind of members can support you? Are we talking about global pharma, other biotech? So you talked before about out-licensing. So let's describe a bit there. So give more insights in that.
So I believe how other members can actually help us is further establishing our region as a huge bioscience and pharma hub. I have an idea in my mind is that we in our region can become kind of a German Silicon Valley. And like at Silicon Valley,
not a European Silicon Valley,
or maybe European. Let's plan for success.
Yeah.
And like at Silicon Valley, each company in our region can benefit, contribute and benefit from this established image. And I believe this is also a possibility to win the so-called war of talents.
Yeah, one of the big topics.
Definitely.
Yeah. So I've seen that we have some questions in the chat, so therefore I was a bit distracted. One question from Umar Khalid. Thank you for posting that. So in the clinical trial, is Heidelberg Pharma also testing combinational therapy options?
Currently, we are not testing combinational options, but I'm pretty sure. So we believe that treatment of cancer, the key is in combination, and probably in future we will consider also combination therapy in the clinical trial.
Perfect. And another question is, so first of all, apologies, she missed first minutes, maybe you have replied, but I don't think so. So what are side effects on animal models of targeting such a fundamental process?
ATACs, we have done a lot of modeling and trials with our ATACs, and I have to say, compared to other ADCs, ATACs have a very clear tox profile. So what we have seen in animal trials was especially liver tox, which comes from unspecific uptake of ADCs, well described and well known toxicity. So unspecific uptake of ADCs by the liver and related kidney tox.
Okay. So I would say last question, because we have still some questions. So Stephanie is not convinced that will be the last. Let me see. So what kind of application can support or accelerate the development of ADCs? In brackets, maybe in the R&D phase, preclinical phase, clinical trial, etc.
Very good question. Application, you mean software? Or what kind of application?
Maybe we can take the next question, and Anugrah, you can specify with application if you have time. Going back, what is the preferred route of administration for the therapies that you have tested in animals so far? So from Victor Diversa.
Well, currently, we are testing intravenous application of the ADCs.
Okay. Perfect. So I don't see at the moment a specification of the previous question. Probably there will be more questions in the chat. We collect them, we send it to you, and we share the answer with the participant. Okay.
Yeah.
Thank you. So I think we had a very nice overview with some more deep insights at Heidelberg Pharma. Thank you very much. Now maybe some questions a bit more about you. So how does a typical day at Heidelberg Pharma looks like for you?
I believe it starts, like for everyone, with a cup of coffee and checking emails. And so here at Heidelberg Pharma, I'm responsible for contracting, for in-licensing questions, out-licensing, and of course supporting our scientists, especially in communication with the outer world. So acting like kind of an ambassador.
Okay.
But also internally. So we have besides the R&D teams, we have also a finance department and the legal department. So I'm building kind of a bridge between those departments and bringing all together, helping them to bring our technology and our company forward.
Okay, so it's a lot of communication.
It's a lot of communication, a lot of emails, a lot of calls.
Yeah, perfect. Thank you. And if you are not in Ladenburg, in the office, where can we find you? Don't say home office.
Probably, yes. But also in Ladenburg, at home. So I live here in Ladenburg, okay. And I think in my garage or in my garden.
Okay, so take notes if you want to find Artyom. I was asking Artyom that he could nominate the next member that could join our M2M series. So you can repeat, sorry for that, who you would like to see in the interview and why.
Yeah, so I would like to see Hummingbird next M&M. Because I scrolled through the list of BioRN members, and I believe that so we have potential great synergy between our company and Hummingbird. We are bringing the therapy, Hummingbird is bringing the diagnostics. Being able of diagnose the cancer at the early stage. And being able to provide more tailored therapy to the patients, it's a great effort. And from my point of view, it's all about the patient at the end.
Okay, perfect. So we will definitely get in touch with some Hummingbird's representatives here in Heidelberg. And yeah, let's see if they will be available. So we are coming to the end of the interview. I would like to thank you, Artyom, for piloting with us this new event. So you see there are still some problems to be solved, but for sure the interview has been recorded and will be available on in our media lounge soon in the next days. You can watch that on demand. You can share it with your colleague. You can come back to Artyom with some meetings. And yeah, thank you to Heidelberg Pharma to hosting us. We have already a next date for M2M. So this will be April 9th with Singleron. So you can tune in at 12. And yeah, join us for the next M2M. Thank you very much.
Thank you very much.
Yeah, bye-bye.